Hello again, everyone. Welcome to Chapter 11, Microbial Genetics. This chapter is going to provide an introduction to microbial genetics and will mostly be a review from Bio 181 genetics as the principles mostly remain the same. Um, Genetic material provides the structure and function of microbes. And most microorganisms, except for viruses, use DNA as their genetic storage material. And we're going to do a review of what the structure of DNA is, how it's packaged in prokaryotes and eukaryotes, and what the essential functions of DNA are. The image you see to the right is a representation of the basic building blocks of DNA, which are called nucleotides. They're made up of three components, generally speaking, and in the center is what we have as our central sugar. And in DNA, this is called a deoxyribose. It's a ribose sugar, which instead of on its second carbon here at the point, instead of having another OH group like you see with its neighbor, it just has a hydrogen. So it's deoxygenated at that second carbon. Next, we have quite simply a phosphate group attached to the final carbon in the deoxyribose. And attached to what we label as the first carbon here, we have a nitrogenous base. And the nitrogenous base is what actually can vary from nucleotide to nucleotide and defines what type of nucleotide it is. The rest of the components here are always going to be the same. Those different types of nitrogenous bases include adenine, abbreviated as A, guanine, abbreviated as G, cytosine, abbreviated as C, or thymine, abbreviated as T. Now, as I alluded to, there are different numbers that we label the central deoxyribose sugar. And where the nitrogenous base attaches to, that carbon right there at this point is always carbon 1. And then we just count in a clockwise direction. So carbon 1, carbon 2, carbon 3, carbon 4, and then this carbon is written out for us nicely. So then that is carbon 5. And the nitrogenous base is always attached, shown with this bar here, to the carbon 1. And the phosphate is always attached to carbon 5. Now, how do these nucleotides attach to each other? Well, they attach to each other through something that we call a phosphodiester bond between a phosphate of one nucleotide and the sugar or the deoxyribose of another nucleotide. Here, if we look at these two monomers, these two nucleotides, this top one and this bottom one, we can see the phosphodiester bond being formed through what's called a condensation reaction. It is called a condensation reaction because it produces a condensate, specifically water or H2O. How does it do that? Well, when this phosphodiester bond forms, this oxygen from the sugar or the deoxyribose binds with the phosphorus group of the phosphate. And phosphorus can only hold so many bonds, and so it gives up its hydroxyl group. And the same is true about the oxygen here, which binds with phosphate. It can only have, in general, 2 bonds. And so it's going to lose its hydrogen, and these two groups will combine together. An OH plus an H creates 2 hydrogens and an oxygen, or our water. And if you look over here to the right, this is where we have what's called a phosphodiester bond. And this is where the phosphate links up with that deoxyribose. Now, something that you need to keep in mind here is where these bonds occurred. So I already described a few of these important carbons to you. The first carbon is where the nitrogenous base binds to. The second carbon, this is actually RNA shown here. If this was a DNA, you would expect no oxygen to be here, and this carbon would be bound to hydrogen. Okay, so that's an important number to remember as well. And the third carbon is where we get the sugar binding to the phosphate. And then lastly, the fifth carbon is where the sugar binds to the phosphate here of its own phosphate. So where the phosphodiester bond links is at the third carbon to a different nucleotide's phosphate, and where the sugar binds to its own phosphate is at the fifth carbon. You see these little apostrophes here. That means in math and biology that we call that a prime. So we might say this is one prime, 2 prime, 3 prime, 4 prime, and 5 prime. It's a 5 carbon a sugar. And what becomes of this? Well, if we string a bunch of these together, it doesn't matter how many we string together. If you look at one end, you'll see one free 3 prime carbon bound to a hydroxyl group. And if you look and follow up to the other end of the chain, you're going to see a 5 prime carbon bound to a phosphate, because this is this little sort of balloon that is attached to the final carbon of the deoxyribose, or the ribose if you have a RNA molecule or a DNA molecule. More on RNA later, of course. So we can string together a whole bunch of these together. So here's one nucleotide, and you can see here's your nitrogenous base. So that must be the first carbon, second carbon, third carbon. And then here's a fifth carbon that's bound to phosphate. So this is kind of a very simple cartoon representation of it. So where do you think that next nucleotide is going to bind to? Which carbon here? Which point? See if you can find it. And I'll go ahead and pull it up now. We would expect that it's this new nucleotide, its phosphate will bind to the third carbon here. And if you look at this nucleotide, where would we expect another nucleotide's phosphate to attach to? Well, of course, it's going to be again to the third carbon. So when your cells build out its DNA or RNA, it always attaches the new phosphate from the new nucleotide to that 3 prime end there. And we can go all the way through and create as long as a chain as we need to. So this portion right here, if you notice, it's a repeating phosphate, ribose, phosphate, ribose, phosphate, ribose, or deoxyribose, depending on if it's DNA or RNA. And this is what we call the backbone, and it's made up of sugar and phosphate. Now, interestingly enough, DNA has 2 strands to it. It's called double-stranded, and those 2 strands are complementary to each other. And what we mean when we say complementary is one strand is built in such a way, and the other strand is built in such a way that they can bind with each other. See these little dotted lines? These represent what we call hydrogen bonds. So that's hydrogens bonding to oxygen, or it could be a hydrogen bonding to nitrogen, would be another example of a hydrogen bond in these nitrogenous bases. And we need a certain shape, and we need these bonds to be in very specific positions in order for that to occur. And the shapes that match up to each other, like 2 pieces in a puzzle, are going to be adenine and thymine, and guanine and cytosine. So these complementary strands, you're always going to find an adenine across from a thymine, and you'll always find a guanine across from a cytosine. So if you look at the rest of this molecule, you'll note that that's the case for the rest of the nucleotides in the 2 strands. And you should always be aware that A's always bind to T's and G's always bind to C's. Now, G's and C's are interesting because they actually have 3 hydrogen bonds that are available to form. So G's and C's are more tightly bound to each other because they have essentially, think of it as 3 hands holding together, instead of what we see with A's and T's, where only 2 hands are able to hold each other or hydrogen bond with one another. In reality, DNA is not flat like this. DNA actually forms what we call a helix. So it's helical in nature, and because there's 2 strands, we call it the double helix. So it looks something like this, where the rungs of this helix here, these outside ribbons, which are just a cartoonish drawing, are again the phospholipase backbone or deoxyribose, depending on if it's DNA or RNA. And in the middle here, pointing towards each other, we have those steps here that bind to each other, guanine to a cytosine and adenine to a thymine. We simplify the hydrogen bonds by just drawing some dots here. But remember, guanine to cytosine always has 3 bonds, and adenine to thymine always has 2. And here's just another representation, again, to drill that, again, how we name our carbons. So we always name the first carbon, the one that binds to the base. And then the second carbon is where it can either be a hydroxyl. So if you look at this, you should be able to identify this as a ribose. So this would be an RNA nucleotide. Would make up a ribonucleic acid, or RNA. And if this was just a hydrogen, no oxygen, then this would be a DNA. In other words, deoxyribonucleic acid. The third prime carbon here is the one that can be used to bind to the phosphate of a different nucleotide. And the fifth prime carbon forms this flag that holds on to the nucleotide's own phosphate group. And this was discovered quite a few decades ago, in about the 60s, by these 3 major contributors. Actually, there's 4 here, but the main ones were James Watson and Francis Crick, observed in the image to the left, and then Rosalind Franklin in the image in the top right corner. And Maurice Wilkins also had his hand in it as well. Francis Crick was particularly important because she snapped the images. And so she was a expert at taking X-ray diffraction images. This uses, instead of electrons, like we've learned about electron microscopy, this uses X-rays to take an image. And this is able to take very small images. And what you see here is an image of a strand of DNA looking from the bottom through the double helix there. And I can't make a lot of sense out of it, but they knew right off the importance of what they were looking at. And they actually, by looking at that image alone, were able to build these complex models of the DNA. You can see, looking at this DNA molecule, these are the phosphate and ribose sugars on the outside here. Here's a ribose here, it'd be a phosphate above here, with the oxygens hanging off to the sides. Up and up we go. And then these rungs here would support those nitrogenous bases, also highlighted here, that connect the two. And they were able to calculate the size and the pitch of these different grooves in the helix and determine which bases were pairing with which. This was quite incredible at the time. Here we go. 1974 is when their first publication came out. I pulled the publication here, and it seems quite incredible. It comes to us very naturally because we're taught this in our introduction to biology courses. But just by looking at this image, they were able to postulate something incredible. At the time, we didn't know where our genes were stored. We had this concept of there was these units of heritability that determined who we were to a certain degree, and that these were passed on from generation to generation. But we didn't know what it was. It was a mystery molecule. In fact, at the time, most people thought it was proteins because they were so numerous and common and complex. But this is an incredible little couple paragraphs that they left off here. It was only a 2-page paper, but it was, fundamentally changed the way we thought about biology. And they said, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Full details of the structure, including the conditions assumed in building it, together with a set of coordinates for the atoms, will be published elsewhere. Okay, so by looking at that image, they thought, well, there's a way that the cell could possibly make copies of this by merely looking at the structure. So how did they do that? How did they determine how this was going to be copied just by looking at the structure? Well, they noticed that these strands were complementary, right? They determined that A's bind to T's and G's bound to C's. They also determined that the strands, in order for this to work geometrically, the strands run in opposite directions. So if we look at the strand on the left, this bottom part, we can see that it has the free 3' hydroxyl group. So this would be called the 3' end. And if we look at the top, this 5' carbon is free from being bound to another nucleotide, so this is called the 5' end. And if we look at the other strand, it's going in the opposite direction. So it's parallel, meaning they both line up in the same plane. They're vertical here. They could be both horizontal together, but they're parallel lines to each other. And this one has the exact opposite, as the 5' free end on this side and the 3' free end on this side. So they're considered anti-parallel. Think of it as a street that's a two-way street. One lane goes one way, the other lane goes the other way. So we have these ends, the 5' phosphate end and the 3' sugar end, that we can take a look at on any strand of DNA. All right. And so what ends up happening is one strand can tell the story of the other strand. So if you have a single strand of DNA and you're charged with trying to build the code for the other strand of K, you can do so by just looking at that first strand. So if you're taking one strand and you're synthesizing a new strand, then we would call that strand the template strand. So here's the template strand. Take a moment and see if you can figure out what the synthesized strand would be. Pause the video here. Okay, so quite simply, we would, beneath that template strand, we could rewrite the new strand, and in a way we're simulating this synthesis here. We know that for C, we'd want to put down a G, for the A, we put down a T, and so on and so forth, all the way across, and we have our new strand. I could also ask you to please, if this was the 5' end, then what end would this be? And you would say that's the 3' end. And if this is the 5' end, you know this is the 3' end. So what would the other strand's end be over here? Well, it would be the opposite. It would be the 5' end. And that indicated to Watson and Crick that there could be a copying mechanism by just copying a template strand into a new strand by the organism. All right, so now that we've talked a little bit about the basic structure of DNA, let's take a look at how it's packaged for prokaryotes and eukaryotes. Let's start with prokaryotes. They have essentially one large circular chromosome. Might look something like this. You know, it's not a perfect circle there. And that's going to be their main genetic information. That's going to store everything that bacteria needs to survive day-to-day things. On the other hand, bacteria have these, and some yeast have it, but we want to really focus just on, prokaryotes here. There's many exceptions to the rule, but bacteria have these amazing extra chromosomal pieces of DNA. Extra means they're not a part of the chromosome, and they're called plasmids. These plasmids are much smaller. This is drawn a little bit large here. It's not really quite to scale, but they're much teenier compared to the bacterial chromosome. They are also circular, so there's no end. It just goes in a loop around and around and around. And they exist somewhere in a slightly different space than the bacterial DNA. And the bacterial DNA is kept in what we call a nucleoid region. An oid, that means like. So it's kind of like a nucleus, but so it's in a specific area, but there's no membrane around it. So it's not a nucleus-bound chromosome. On the other hand, eukaryotes are much different. They actually have a nucleus, which is just a membrane. We can see kind of shown here, zoomed in, that stores that DNA. And also, if you look at the DNA, it is linear. So it has one end and another end to the other side. So it's not completely circular. And also, eukaryotes have a lot more DNA. And they have to be experts at packaging this DNA. And so what they use are little tricks here. So one thing is they can coil them up. And if you've ever taken a rope or a string or a cord or a headphones or something like that, and if it gets too many twists in it, it makes this super coil back upon itself, and it makes these coils on top of its coils. And that's one method of helping them package it. And they also package those coils up around something called histones. Histones are little proteins that you can wrap that genetic information together so that it doesn't get tangled when you put it into a condensed form called chromosomes. And so you can condense it down into a very condensed form here. Now, this form right here, it's not very readable, but it is packageable. And so chromosomes will form in order to go through cellular replication through a process called mitosis or meiosis. But in general, we have to keep it in this loose form when it's time to go through our different parts of regular cellular life, so that the cell can get in here and read this genetic code. Okay, so what are the essential functions of DNA? Well, for one, it's very important for inheritance. So it's a code book that can be passed from one offspring to from one generation to the next. The other thing it's really important for is for storing information in order to execute certain functions or to create certain structures in the cell. In order to be a form of inheritance, it has to be heritable, right? You have to be able to copy it into a new book and then give that book to a new generation. And that is through a process called DNA replication. And in order for it to be able to provide information for functions and structures, what it is used to make are proteins. So DNA very specifically stores information in it to make proteins. It's a recipe book for proteins. And this process of converting the DNA code into proteins it's broken up into two steps called transcription and translation. Okay, so let's go over how inheritance works first. We'll talk about how DNA is replicated in the next several steps. The protein helicase begins the process of DNA regulation by binding to a specific site on DNA called simply the origin of replication. Helicase has the ability to separate the two strands of DNA at the point of the hydrogen bonds between the bases on either side of the strand. After it is done so, you can think of those free nitrogenous bases as being like Velcro. Once the helicase goes through, if they touch each other, they will easily reattach to one another. And therefore, the cell recruits another protein, and these are called binding proteins or single-stranded binding proteins. They attach to the single strands of DNA, and by doing so, they prevent that DNA from reattaching to one another. Now that we have our two strands separated, it's almost as if we opened up the book and we're able to now read those nitrogenous bases that are found on the inside of this double helix. And the third step involves an enzyme called primase. Primase are these enzymes that can land on single strands of DNA, and they can read the DNA and write a new piece of genetic material. However, they don't write it in the language of DNA. They actually write it in the language of RNA. RNA is different from DNA in that it's typically single-stranded, and instead of being made of deoxy, which is what the D stands for in DNA, it's made of just ribose instead. So it still has that OH group at the second carbon in the ribose. Now, when you're getting familiar with both DNA replication and transcription, which will come later in this chapter, just keep in mind that all of these enzymes they have to drive a certain direction on the street. So these primases are no exception to that, and they will always drive from 3' to 5'. So from the 3' end this way down this way towards the 5' end of the street. So we would expect this enzyme to move towards this end and this enzyme to move towards this end, towards the replication fork. See how it kind of makes a fork here? We call this the replication fork region. As it is moving along these strands here, it's reading them. And this gets back to Watson and Crick, who, by looking at this complementary nature of DNA, they suppose that that could be a way that this DNA could be copied. If this primase reads a G, then it will naturally know that it needs to build a C across from it. So as it's moving down this strand, or as in our analogy, the street, it's going to actually be constructing the other side of the street that's complementary to it. So let's go ahead and take a look at what that looks like here. So again, the enzyme is going to read from the 3' direction to the 5' direction. And this top one's also a 3' the 5' direction. And remember, we said they're not just complementary, these strands, to one another. They're also anti-parallel. So if you imagine you're writing the other side of the street, you're writing not 3' to 5', but you're writing that new strand, that new side of the street, 5' to 3'. So 3' to 5' is the way it's read, and the other strand that it produces, the newly synthesized strand, is going to be synthesized 5' to 3'. And so it's going to build across, and there it's going to build a short little piece of RNA, and we call this the primer. It's going to prime the well of DNA synthesis. So it starts this process out, and that's what's unique about primase. There we go. We have our RNA primers. And just a short little review to compare DNA to RNA. Here's our DNA. We've talked about DNA quite a bit. It's a double helix. We have our ribbons here that represent the phosphate and deoxyriboses that are linked together. And if we then jump over here and we look at the RNA, RNA is not made of deoxyribose. It's made of ribonucleic acid. And so the sugar does not have a OH group. It does not have an H group at the second carbon. It has the OH group at the second carbon. It's just plain old ribose. It's typically single-stranded, as shown here, whereas for DNA, it's going to be double-stranded. And there's one more important feature that you need to know about these two, and that is we covered the 4 nitrogenous bases of DNA: cytosine, guanine, adenine, and thymine. And the first 3 are the same for RNA: cytosine, guanine, adenine. However, the very last one, thymine, it doesn't exist in RNA. Rather, RNA uses a uracil as in place of thymine, and that's shown by the letter U. So while in DNA, you're going to find that an adenine always pairs up across from it with a thymine, that's not the case for RNA. When that primer is made, an RNA polymerase reads an A, and it wants to make the match across from it to make that primer, it's going to put a U there instead. Here again, we can see the difference in the way the sugar molecules are built. Here's the first carbon, here's the second carbon, and in DNA, we have deoxygenated that second carbon, so it doesn't have an OH group, it just has a hydrogen present. But if we look at our RNA, it's still just a regular ribose sugar, so it's oxygenated here, that's 2' carbon. And this just shows you how that base pairing is different with DNA and RNA. So for DNA, you see nice and neat, the A's go across from the T's, T across from the A, T across from the A. But if we look at our primer being built using a template strand, we can see that while there's a T for DNA, and we'll see an A across from a T here, whenever there's an A for DNA, our primase is going to put a U there because if you notice, there are no T's in RNA. Okay, wonderful. We have built our primer, and that laid down the foundation to start the process of making these new strands. And it also showed something very unique about the way these two strands are built. If you take a look here, this primase is working towards the replication fork, towards that helicase. Whereas if you look at this one, it's going away from the helicase. And this is going to create a problem, which we'll see very soon here in the production of the two newly synthesized strands. Because again, that primase has to drive 3' to 5'. And as it's doing so, it's building the other side of the street, which is anti-parallel in the 5' to 3' direction. It sounds insignificant to you, but it's important. Imagine if you went to a foreign country and you drove down the wrong street. You'd be in a lot of trouble. So remember that. Don't just glance over this component of the lecture. It's going to be very important as we move forward. So our primers have done their work. Our primase has done its work. It's built those initial primers there. And just keep in mind, all of these enzymes we're going to talk about read 3' to 5' and build 5' to 3'. So as time goes on, that replication fork opens up further with helicase continuing to go down the rails here and spread this double-stranded DNA molecule into its two single strands, held open with those single-stranded binding proteins. And now we have DNA polymerase III come into the picture. And this is going to be the real workhorse of this process. It, like primase, also synthesizes a new strand of nucleotides. However, it does so with DNA nucleotides. So finally, we're starting to build it in the code that we need to build it if we want to replicate this DNA and if we want to pass some DNA to the next generation. But DNA polymerase III, it needs a primer to be present. That's how it knows where to land. And so that's why that primase enzyme is a step before we get to DNA polymerase III, because it gives that landing pad for DNA polymerase III. DNA polymerase III is going to land onto that primer. And I want you to just take for a moment here and think about it. Think which direction you would expect this DNA polymerase III to go and which direction you would think this DNA polymerase III would go. Go ahead and pause the video if you need to and assess your knowledge here. Well, what we should have thought is that this one shown on this top strand, in the cell there's no top strand, but as it's represented on this PowerPoint, it needs to travel in the 3' to 5' direction of the template strand. So it's going to go towards the replication fork, just as we saw with a primase building the primer. And this bottom one is heading in the wrong direction again. It's heading away from the replication fork. And they're going to build across and they're going to synthesize DNA as they go along. So they'll read the base, and then they're going to generate the new nitrogenous base that matches it across the way. All right, so now we've got our first pieces of DNA laid down, and DNA polymerase III can also proofread. So it checks its work and makes sure it doesn't make any mistakes, and it can fix any mistakes along the way. So it's got an eraser attached to that writing utensil as well. So let's just imagine, if we may, that this continues to go on. So DNA helicase is unzipping some more, and our DNA polymerase III that's shown on that strand at the top, it's going to be leading the race because it's moving in the right direction. It doesn't have to start over. It just keeps chugging along, and it's going to synthesize DNA continuously, just a little bit behind that helicase there as that single strand becomes available. But the issue is with this bottom strand here, it's actually going to either come to the end of the DNA strand, or it's going to, and it might just fall off there, or it's going to butt into a new one. And so it may have to wait for a primase to land, create a new landing pad, the primer there, and then that DNA polymerase III can then land on that landing pad, and it's still going to build away from the strand. And so there's these extra steps, and it's going to take a little bit of time. And so therefore, we call the strand where the replication is occurring away from that replication fork, we call that the lagging strand. And the top one we call the one that's working towards the replication fork, we call the leading strand, because it's always going to be a little bit ahead of that bottom strand that has to wait for a new primase to be lay down the new primer, and then the DNA polymerase III can come in and land and generate that new strand of DNA. And we'll talk a little bit more about how we fill in these gaps here, but keep in mind now that we have two unique synthesized strands. We have a leading strand and we have what we call a lagging strand. And these little chunks of DNA that lagging strand create are named after the scientist who discovered them in Japan. His name was Dr. Okazaki, and so we have the Okazaki fragments here. Again, the one that's made continuously towards that replication fork is called the leading strand, and the one that's made in segment by segment is a little bit behind, and we simply name it the lagging strand, and it contains these Okazaki fragments. Why are these fragments? Well, as we said, DNA polymerase III can only build in the 5' to 3' direction. And when we say build, if you think about it fundamentally, if this is a sugar here, right, or this is say this is a nucleotide here, and we have an open 5' end, that means there's a phosphate free here. And before we build the rest of it, let's just focus on what we have. Or I'm sorry, we're in blue here. There's a nucleotide here, and there's the phosphate on one side and the ribose sugar on the other side. And when we say build, what is it doing? It's adding those phosphodiester bonds. So it can add a new nucleotide that binds its phosphate to the old one's ribose and continue on in this direction until we get to the very end. And then you'll have since this is the 3' end, you're going to have a free ribose here. And what DNA polymerase can't do is it can't take that ribose and then bind it to a new strand, the strand that's already here. It can only add free nucleotides to that end. And so you get a gap. This is just a phosphodiester gap where it hasn't bound one ribose sugar to the other nucleotide's phosphate. It can't combine those together, and so we get these gaps or these fragments of the DNA. So that was DNA polymerase III. Now DNA polymerase I gets involved. And students, one of the most common errors I see is confusing 3 with 1, because 3 comes before 1 in this situation. 3 comes before 1, and it lays down that DNA before DNA polymerase I comes along. And why is it called 3 then? Why didn't we call that one 1 and we call this one 2? Well, DNA polymerase III was the third DNA polymerase discovered, and we just named them according to their discovery. So DNA polymerase I, while it comes later in this process, was discovered before 3. And what does DNA polymerase I do? Well, it has a very specific and small job. Its job is to replace those RNA primers and fill them in with DNA. Pretty simple. Now let's get back to these little fragments here on the newly synthesized strand. Sometimes you'll hear people call them nicks, because it's as if the phosphodiester bonds were nicked there. And we need to bridge those together. And a term that you'll see in science for recombining something is ligation. And so we have the enzyme that's named after that process called ligase. It only has one job, and that's simply to connect the ribose, the 3' carbon ribose on one end, and do a condensation reaction, combining it to the phosphate group on the 5' carbon of the other end. And so it combines that together. There we have our ligase. and we've combined the two together. Now I have this little video here. We're going to jump over and watch it. We'll show what that process actually looks like. This video is going to specifically show DNA replication involving the enzymes helicase and DNA polymerase 3. It's going to also show the leading strand and the lagging strand. This is our DNA helicase. It's separating into two strands, single strands, one here and one here. The strand on the bottom has a DNA polymerase 3 moving from the 3' to 5' direction of this single strand and making a copy just right towards that DNA replication fork. And on the other side here, you can see the DNA polymerase 3 has to work the other way. It hit the end of a prior Okazaki fragment. It lets go, and then it comes back and grabs again and starts over, working away from the DNA replication fork. And you see this giant loop means it is always lagging behind the DNA polymerase 3 that's now on. Okay, so that wasn't the best of audio there, but what I was trying to say, if you look at the image, there's a DNA helicase in the center that unwinds the two strands. There is a DNA polymerase 3 at either end, and on one side it is moving towards the replication fork, and on the other side it moves away from the replication fork. Here is a link there if you would like to type that up, and you can go watch the full animation video. I tried not to show too much of it so that we don't have a copyright violation that occurs here. And that process of creating one new synthesis strand per one old synthesis strand means that we have this interesting phenomenon with the two strands, in that one of the template strand, is old for each one of these newly formed double helixes. And because of that, then scientists realize, hey, this is actually a conservative process. We don't just throw away our original template. It's semi-conservative. So half of it is old and half of it is new. And so that's another term that scientists use to describe this process. All right, so that covers as much detail as we're going to cover for the inheritance portion of DNA's functions. Let's dig in now into transcription and translation. So cells have this process of taking DNA, they turn it into a RNA molecule, and then that RNA molecule then goes to the ribosomes and makes DNA from it. And these two steps are called transcription and translation. Transcription is simply the conversion of that DNA template into a complementary RNA molecule. And translation is defined as taking that RNA molecule and taking the code that it's written down in and converting that into a protein. And this is called the central dogma of DNA, of biology. Taking DNA, making a transcript RNA molecule out of it by copying it down into RNA, and then taking that transcript to a ribosome and having that ribosome synthesize some protein by using the code. Let's take a look at the transcription portion first. Transcription relies on essentially just one enzyme, and that's called RNA polymerase. As you can imagine, what it does is it makes a polymer of RNA. And it can, unlike what we saw in DNA replication, it can do a lot of these things all by itself. It doesn't require an RNA primer, so there's no need for primase. It can land on any strand of DNA. All it needs is a specific address, which is just a series of particular nucleic acids in a row that identify it as a place to land. And that is called the promoter region on the DNA. And it can also unwind the DNA as well. So it doesn't have to rely on a helicase to unwind the DNA strand. And it can read the DNA strand and create that complementary message in RNA, and that's what this image is showing here. Is this RNA polymerase is this background glob here, and it lands on that RNA promoter region, unwinds the DNA strand, and creates that new mRNA molecule. Notice there's U's here and no T's being generated. And now it's got a copy of this transcription unit that it can take to the ribosome. Now, before that mRNA can go off to the ribosome in eukaryotes have a lot of different versions of each gene, and they carry something in between them called introns. And introns need to be cut out depending on which version of the recipe of that particular gene it wants to make. In other words, they're excised out. So we have these proteins that can land there, and they form this what's called a spliceosome, and they cut out those introns and they stitch together the parts of the DNA or RNA that they want for that particular version of the recipe of a protein that they want to make. That is called splicing. So again, the introns are cut out, and the exons, or the red portions shown here in red, are spliced back together to make one contiguous strand of mRNA with all of the correct ingredients for that version of the protein that the cell wants to make at that time. And it can be hard to memorize. Is it the introns or the exons that get spliced back together and then go to the ribosome? I like to remember it by thinking that exons will exit the nucleus to be translated into proteins. All right, so now we have made our transcript called RNA, and now it's time to move further along in the process and do our translation, where we take that copy of the code in RNA and we convert it into protein. And briefly, let's just remember that proteins are a series of amino acids linked together in a chain. So just like we saw with DNA being made up of monomeric nucleotides, proteins are made up of the monomers amino acids. There's a lot more amino acids than there are different nucleotides, as we can see here, and they have different qualities and traits about them. Some of them vary in the types of what we call the R group, or the side chain here. Just like we saw with nucleotides, where they all have a phosphate group and a sugar group, and if they're DNA, it's a deoxyribose. If they're RNA, it's a ribose sugar, and those never change between each nucleotide. Only the nitrogenous bases vary and determines what kind of nucleotide it is: an adenine, thymine, guanine, cytosine, or uracil. In the example of amino acids, they are amino acid, means that they have an amine group, which is this NH3, and they also have an acid group, which is this carboxylic acid group here to the other side. And what you need to pay attention to differentiate these different amino acids is the group, the side chain group here. The most simplest amino acid has a side chain that is a hydrogen, and some of them can get a little bit more complex, as we can see here. And these side chains provide different traits. If you do not remember what polar and nonpolar mean, or electrically charged, you better go back and review the basic biomolecules of life lecture, as I recommended earlier in the semester. But you can have groups of amino acids that are nonpolar, or that are polar or electrically charged, and that determines how they interface and interact with the world, as well as within the same amino acid chain that makes up the protein. And so what do we need to have for ingredients in order to go from our messenger RNA and in order to generate our protein? Well, let's take a look at that. First, we need our RNA. It's called a messenger RNA, or mRNA, because it sends the code that it copied from DNA. It sends it over to the ribosome. And we need amino acids in order to create our new strand of our new protein molecule. And those amino acids are delivered through this molecule called tRNA. It's also an RNA molecule, and it's kind of shaped in the shape of a T. And at one end, it's going to have our amino acid, and at the other end, it's going to have a code translator. And we need our ribosome. And interestingly, the ribosome is actually made up of two things. It's made up of proteins and RNA. This RNA doesn't get translated into protein. It just stays as RNA, and it helps in the structure and function of the ribosome. See, ribosomes are these very large, complex combinations of RNA and protein glued together, and they help facilitate reading the code of mRNA and turning it into the amino acid sequence of whatever particular protein that mRNA codes for. tRNA, kind of think of this as kind of our decoder. tRNA is a bilingual molecule. It carries that amino acid on one end here, and then at the other, towards the other end of this RNA, you see the nucleotide bases here written out by their first letter. It contains the decoder, and that is called the anticodon. So mRNA carries a message on what amino acids to add to make a protein. And they're stored in these little codes that are made up of 3 letters. In our language, we have words that vary in sizes, but in genetic language, all words are 3 letters long. And that anticodon of that tRNA, if it matches a particular word, we know that that word translates for its particular amino acid. And so each tRNA only has one anticodon. And therefore, cells have to have tRNAs for every amino acid. And so they have their own specific anticodon that will match that amino acid. Humans have devised a codon chart. This is how we can do it. And in this codon chart, we see that all the words are grouped into groups of 3. And by looking at a sequence, we can decode it by using a codon chart. You start with where it says the first letter, and you find whatever that first letter is in the codon. Let's say it's an A. And then you look at the top here for the second letter, and let's say the second letter you saw in that mRNA was a U. So then you would find these two where they intersect, and it's going to be one of these 3 options here, or 4 options. We'll talk about start codons in a moment. And then lastly, you're going to keep track of that box. You're going to come over here and look at the third letter and see, okay, maybe it's a C. So an AUC is going to code for an isoleucine molecule. And that's how you use a codon chart. Here's an example of an mRNA strand here and they don't separate them out into 3 letters. You have to do that on your own. So there's no spaces or anything. This AUG is the first codon, this GUG is the second, CAC is the third, CUG is the fourth, and ACU is the last one. And each one of these words is going to be able to bind to a specific anticodon and release a specific amino acid. All right, so let's go ahead and go through our naming conventions of these different strands and different components. Now, here I have an example of a double strand of DNA. And you'll note that here at the bottom we have the template strand. So this is the strand that's going to be used by RNA polymerase to make our mRNA strand. And our RNA polymerase is like all the other enzymes we talked about. It can only read in the 3' to 5' direction. So we know that RNA will land over here and it's going to build in this direction. So here's our mRNA strand. This coding strand, what does coding strand mean? Coding strand means that it is what the code will look like when it gets sent to the ribosome. And this coding strand is complementary to the template strand, just like the mRNA strand is complementary to the template strand. So notice how the letters are going to be the exact same between the two, with the exception that because this is RNA, Instead of having a thymine, it has a uracil there. So that's why we call it the coding strand. It is presenting the code the same as it would be presented to the ribosome. Now I've labeled some features here for you to keep track of as well. One of them we've already talked about is the promoter region. The promoter region is what identifies that DNA strand as somewhere that our RNA polymerase should bind and make our transcript from it. And these other components we'll talk about later. There's going to be a ribosome binding site, which allows the ribosome to know where to attach for this particular transcript. And then there's going to be a start codon, and that will tell the ribosome where the first amino acid is for the new protein. And there's going to be a stop codon. And this codon tells the ribosome where to stop making protein. So it is only going to make a protein starting from here to here, and it's going to ignore the excess material off to the left and off to the right of the start codon and the stop codon. Okay, so we can break up these codons again. Here I've drawn lines in between the two to show how they'd be broken up, and we can go ahead and use our codon chart. I recommend, if you'd like to pause this video, write down this code and see if you can decode it and see if you understand it well enough at this point that you can do it on your own, and if not, where you went wrong so you can make the proper correction. So please pause it and see if you can do it along with me. So we've broken this code up here. And now I'm going to use the codon chart, and AUG encodes for methionine, GUG valine, CAC histidine, CUG leucine, and ACU threonine. Hopefully all of you got those correct. If not, take a moment, pause, and see where you made your error there. And that methionine I want to draw your attention to right here is the start codon. It's the only one that I require students to memorize because it's so important that it starts where the gene is turned into a protein. And that start codon series is AUG. If we looked at the coding strand of the DNA, it would say ATG. For stop codons, there's actually a few different ones that can be used, so I don't require you to memorize those, but be aware of AUG or ATG. That should tell you, okay, there should be a gene starting here. All right, so this little GIF here shows you what that translation process looks like. Again, that big nebulous glob in the background. In this situation, that's supposed to be your ribosome. And this long single strand right here is that mRNA code. And you can see that that's the tRNA. And this is its anticodon. And if that anticodon matches up with the codon of the mRNA, then it's more than happy to release its amino acid. And the ribosome knows exactly what to do with that amino acid. It attaches it to that growing amino acid strand, making the protein. Now, it doesn't really look as clean as this. What's going on here is, lucky enough, the tRNA, the right tRNA is always going in. But in nature, it's not like that. tRNAs are flowing in and out, and they're seeing if they have a match. If they don't, they pop back out of that region. And if there is a match, then they stay attached and release their amino acid, and then they fall out the other end. This slide here summarizes the differences between the prokaryotic and eukaryotic transcription and translation process. In the light blue, we can see our DNA strand here is circular. It is taking up a little bit too much room in the cell, but it is what it is. Whereas when we look at the eukaryote, we see this linear strand of DNA. And we get a transcription product, which is our mRNA. And at this point, we call it pre-mRNA because it hasn't yet had a processing done to it, such as the removal of those introns. And once those introns are removed, there's some other things that happen that I don't require for this course, but it is now prepared to leave the nucleus. And if you look over at the prokaryote, there is no nucleus. So it doesn't require this. There's no pre-mRNA. It's ready to go straight over to the ribosome. There's no introns to remove. And after it leaves the nucleus, then it can start interacting with the ribosomes. The ribosomes can look at that mRNA and start generating the proteins from it that are encoded there. And that's not the case with the prokaryote. There is no need to wait until it leaves the nucleus. It can start building right away those proteins through the process of translation. Now, there are a lot of genes that code for proteins in cells, and just bacteria alone, we can see that E. coli, which is the most well studied, so we've identified the most protein coding genes there, but we're already up to 4,300 different genes that protein coding genes that we're aware of in E. coli's DNA. And eukaryotes, there's much, much more than that. And E. coli doesn't want to have all of these genes on all at the same time, all at the same rate. So just like enzymes work to control the level of enzymatic activity and control the reaction rates of chemical reactions throughout the cell also needs methods in order to control what genes are turned on and when. Now, that's not true for all genes per se. Some genes are just continuously expressed. We don't want to foul up their normal expression, and they're just part of normal cellular operations. And so you need just a certain amount of basal expression all the time. And those are called constitutive genes. Constitutive genes, they're always continuously expressed. In other words, they are transcribed and then translated into proteins. And that process, that completed process, we call expressing of the genes. Then we have facultative genes. Facultative genes are only expressed when we need to use them. And this requires certain stimuli in order to turn those genes off or to turn those genes on. And a good amount of genes are constitutive, about 60 to 80% of them, and the rest are facultative. So let's take a look at how this works. Well, we already discussed the promoter region of DNA. These are special sequences where RNA polymerase knows where to bind and initiate transcription. And that would be called the control region. It has another component to it. If it is facultative, there might be an operator attached to it. That operator is attached to the promoter and acts as kind of a traffic light for RNA polymerase, allowing RNA polymerase to transcribe through that region, or it might get halted and prevented from copying the rest of the material. And altogether, this might have several genes actually under control of the same promoter and operator. And together we would call this control region and the structural genes an operon. So you can imagine it looking something like this. Here's our RNA polymerase, and it saw the promoter, it landed, it attached to the promoter region, which is just a specific sequence of nucleotides. There's nothing else there. There's no primer, there's no other proteins. They're necessary for the promoter to function on that RNA polymerase. You can have transcription factors that help it land, but for all intents and purposes, we have our promoter, the RNA polymerase is bound, and now that operator appears to be a red light right now. It's off. And so that cyber truck, that RNA polymerase, does not go down the raceway here. It does not look at that template strand and make an mRNA copy. But under certain environmental stimuli, that can turn to a green light, and it can go ahead and copy the way through. One of those sets of genes are for the production of lactase. Lactase allows for the degradation of lactose, a common milk sugar. And you can imagine if you're in a coli, or if you're a person too, you don't always get exposed to milk sugar. And so you don't want to be spending resources on making this this enzyme here, but you're going to turn it on in the presence of those sugars. And we can see there are several different genes involved in this. I won't get too much into the weeds about this particular operon, but we see now, and when that sugar is around, then your body starts getting to work, or the E. coli starts getting to work to transcribing that lac operon and transcribing these different genes so that they can be translated. And at the very end here, there is probably going to be a terminator. A terminator is like the opposite of a promoter. A terminator tells the gene, the RNA polymerase, to stop because it doesn't want to spend any more resources than it has to making more, copying more stuff over. So again, we have these operons, which are a collection of genes related to usually all related to a similar metabolic process, and they're all controlled by the same promoter and operator system at that control region. We have two types of facultative genes we need to be aware of. One are inducible operons, and inducible operons have to be induced to make the transcript. So they're normally off. And when the environmental stimuli comes in, then it turns it on. And then we have repressible operons, and these ones are normally on, but if something from the environment comes in, and that stimuli occurs on that transcript, it represses it, and that's why it's called repressible. So inducible operons are normally off, repressible operons are normally on, and the environmental stimuli turns inducible operons on, and environmental stimuli turn off repressible operons. And there are a lot of these enzymes involved. This is just a little short snippet of galactose metabolism that need to be turned on or off depending on what sort of product is brought in. So it can control a lot of different enzymes, and one of those enzymes is the lac, is found within the lac operon, the lactase. There's 3 different structural genes that are involved in this process. One, the most important of all of them, is LAC-Z, and this encodes for the protein beta-galactosidase. Beta-galactosidase is the enzyme that metabolizes lactose. Then we have Y, and Y is translates for lac permease, and lac permease is a permease that goes to the cell membrane, and it's a protein, it's a transmembrane protein that allows lactose to be transported into the cell. Of course, we want to start collecting that lactose. And then finally, we have LAC-A, which is, it's called a transacetylase, and this can be used to metabolize other disaccharides that you might find maybe lactose within. So we're not going to get too much into the weeds there, though, but you have LAC-Z, which is the enzyme that metabolizes lactose, LAC-Y, which is the permease, and then LAC-A that can help out with other disaccharides that might be present. Okay, so the lac operon is an inducible operon. So we would be able to expect that this is normally turned off if I told you it was an inducible one. And when the right stimuli comes in, it will be turned on. And how does that work? Well, with inducible operons, that means right at that operator, there's some red light. And so there's actually a protein that's in the way here. So these proteins are called repressor proteins. This is encoded by LAC-I. This repressor protein binds to that operator, and it blocks that RNA polymerase from getting through. And this is very typical for what you would expect with an inducible protein. And so if someone could imagine what's going on when we have a stimuli here, is we could imagine that that's going to be, that stimuli will remove that repressor protein. And we have allolactose. This is a derivative of lactose. So if lactose is floating around, let's say in your digestive tract, E. coli just kind of picks some of it up naturally. A little bit will get picked up by the cell, and it will create some allolactose as a consequence, and that allolactose will bind to that repressor protein, change the shape of it, and relieve that operon of that repressor protein. And what happens? The RNA polymerase is going to go through and make a transcription copy of that region. So that's the only version of an inducible system I want you to be aware of, is LAC is a inducible operon. Okay. Now the TRP operon is a repressible operon. TRP stands for tryptophan. This is a essential amino acid. Okay, so you can imagine it's something that generally needs to always be made. So that's why it's a repressible operon. So under normal conditions, the cell needs to be making this amino acid tryptophan. And under certain conditions, it might not want to produce that tryptophan. So it's an essential amino acid. But if there's plenty of tryptophan around, then we want to now halt synthesizing it. Why waste our resources on something that's plentiful? And therefore, this is a repressible operon. So that action is going to actually trigger this inactive repressor to, when tryptophan is present, it's going to bind to that repressor protein and make it in the correct shape to land on that operator and block RNA polymerase. So that last little bit there was really only the new material that you probably haven't already seen before. I'm talking about inducible operons and repressible operons, and that wraps up our discussion for microbial genetics. I hope you found it to be a good review and add a little bit extra to your knowledge there. Thank you so much for tuning in, and I hope to see you in the next one.